Hi everyone, so this is our first lecture. I'm going to talk about DNA structure, more like nucleic acid structure, and the different forms that nucleic acids such as DNA and RNA come in. And we'll get into some of those details because we really need to understand the basics of DNA and RNA and how information is actually stored inside of a cell. So let's get to it. First, nucleotides. So you'll hear nucleotide as a term thrown around a lot. Now, sometimes you'll hear nucleosides versus nucleotides. When I was doing HIV work, some of the drugs that we would talk about were nucleoside analogs. So they were artificial nucleotides, or not nucleotides. They lack a phosphate attached to the sugar. So let's talk about the structure a little bit. You've always got your base here. And your base can be A's, T's, C's, G's, or U's. I'll talk about that more in the next slide. And then attached here is a sugar. And then off of that sugar, this little ribose here is going to be, that's a nucleoside, okay? Just your base with your sugar. Now, if you attach phosphates here, we start calling those nucleotides. You can have a couple phosphates attached here. You can have one, but that's the basic of a nucleotide. So we're usually talking about this. When I say a nucleic acid or a nucleic acid base or a nucleotide, that's what we're usually referring to. Nucleosides actually don't typically come up too often unless you're talking about generating DNA, some of the pathways that actually make these, which is more of a biochemistry angle. But yeah, so that's what this is. And then we have these nice bonds here. There's a glycosidic linkage here. And we have a nice phosphodiester bond, which I'll talk about in another slide in just a moment. So let's kind of keep moving on. But that's our basic nucleotide. So I mentioned there's cytosine, guanine, thymine, adenine, and then RNA has uracil instead of thymine. Those are all the naturally occurring nucleic acids. Now, there are some synthetic ones that we can make. Typically, those are used in medical applications. Like I mentioned, with HIV drugs, those would be synthetic nucleosides or nucleic acids that we have to generate. Now, what's really interesting about DNA is that it has directionality. And the directionality is really, really important because it's going to keep coming up as we talk about DNA replication. So that's making more DNA. As we talk about transcription, which is taking a piece of DNA and converting it into a complementary piece of RNA. And it's also really important when we talk about translation, which is when we take our RNA and translate it using a ribosome and transfer RNA into a set of amino acids that make proteins, which then do things. So it's really important that we understand directionality. You're going to hear 5' and 3' thrown around all the time. It used to drive me crazy. It wasn't explained to me super well at the beginning, so I'm going to try and explain it pretty well here. So if we look at our basic nucleotide, so we'll get cytosine up here. We've got our base, we've got our ribose, and I labeled the carbons here. So we've got 1, 2, 3, 4, 5 carbons. And on that 5th carbon is going to be that phosphate. So if I back up here, here's our 1, 2, 3, 4, 5 carbon. There's our phosphate attached. So there's our phosphate. And so that is the 5' carbon that phosphate is attached onto. So therefore, this is the 5' end. Hopefully that makes sense. So if that's the 5' end, well, let's see. If there's 5' and 3', let's see down here. So here's thymine, there's our base. There's our ribose, our nice little sugar there. And if we just, pull these down, 1, 2, 3, 4, 5. Okay, so there's my 5' carbon has a phosphate, but in this case, it's already attached to something, so we don't really worry about it. There's a 5' sticking off, 5' phosphate sticking off the end here, but this one it's attached, so we don't worry about that. But on this end, we've got our 1, 2, 3', and on our 3' end, we've got this hydroxyl group sticking off right here. Okay, so 1, 2, 3, there's our hydroxyl group. Okay, so that's when we don't have anything attached here, that's where we're going to attach that new phosphate. When we bring another nucleotide in and stick it right here, that's where we're going to attach it to that 3' hydroxyl group. Nice and reactive. We're going to be able to generate a water from it. I'm going to talk about that in the next 2 slides. Okay, but that's where that nomenclature comes from. So we have a 5' end and a 3' end. The most important thing that you can remember, because it can get a little wonky when we start talking about polymerases and the directionality of polymerases, especially the directionality of polymerase fact checking, or maybe fact checking is the wrong word, polymerase back checking. So as they build new DNA molecules, the DNA polymerase will double check its work and make sure it's doing a good job. At least in higher order organisms, they tend to do that. And so we need to understand directionality because there's a directionality to how they actually fact check themselves or to how they actually add on new nucleotides. And the best thing to remember is 5' to 3' always is how we build. So we start at our 5' and we're going to build directionally towards our 3'. So this 3' hydroxyl is going to get another phosphate attached here. So if I'm doing replication and I want to make a longer DNA molecule, I'm going to take a C, a T, an A, a G, whatever, and stick it here. And I'm going to keep building towards this direction. Because as I tack this guy on, well, now I've got the 5' end of that new nucleotide is kind of hidden now. So it's trapped here, and then I've got a new 3' end. Okay? So that's how the directionality of DNA works. Now, in DNA, there are 4 nucleotides. There's C's and G's and T's and A's, and they match up. So cytosine and guanine match up. They form 3 hydrogen bonds. Thymine and adenine also match up, and they form 2 hydrogen bonds there. Now, the bonding is really, really important. I'll get to that in a little while. And you'll notice guanine cannot bind to thymine, and cytosine cannot bind to adenine. Okay? We can't have this cross-reactivity here, if you want to call it that. That's probably not the right word. They're not going to form bonds. I say that. So you can have mutagens come in and alter this binding. You could have synthetic nucleotides that actually can allow this kind of bonding. When things go wrong is when you could get maybe a cytosine bound to an adenine. And that would be really strange because then when you try and replicate, your DNA, your polymerases don't know which side is the right side. So it says, well, it's supposed to be cytosine and guanine, so I need to make a new cytosine and guanine here, but I have a cytosine and adenine. I don't know which one to make. So the 2 daughter strands could then, one could have an adenine and one could have a cytosine. It's going to be really messed up and weird. Anywho, so one other thing here. So we have directionality. Now, DNA, as we've seen, and I'm going to show you in more pictures later, it has 2 sides. It has 2 strands, right? That's a pretty commonly understood concept, but here's where it comes from. So we have this one strand going this direction, 5' to 3', right? There's a 3'. And then the opposite side, it goes the other direction. So we have our 5' down here heading towards our 3'. And so you get 5' to 3' on one side, 5' to 3' in the other direction. And they're moving in opposite directions. Now, something that does come up sometimes is what about RNA? So in RNA, they have C's, they have G's, they have A's. They don't have thymines, but it's okay. The trick to RNA is that the difference between RNA and DNA is so small. There's only really 2 big differences. In RNA, the thymine is replaced with a nucleotide we call uracil, but all that really happens, this carbon here, this methyl group attached here, goes away. And so now it has become uracil. So that's one of the big differences. And then the other big difference is that RNA has a hydroxyl group attached here. Okay. So, and in fact, let me just jump down here. So on the next slide here, we have DNA versus RNA. So yes, uracil itself has a different structure than thymine, though it's super, super similar. DNA versus RNA, the big difference that we see is we lose this hydroxyl group and we're left with just a hydrogen here on that 2' carbon. I added these numberings here. So we've got 1, 2, 3, 4, 5 carbons, 1, 2, 3, 4, 5 carbons, still the same, almost identical, but you see we have a hydroxyl group on our RNA, which is ribonucleic acid. This is a ribose. And if we take that oxygen off, we deoxide it, deoxyribose, thus deoxyribonucleic acid. So that's where that name actually comes from. Now I want to talk a little bit more about how we actually form DNA. So it's a condensation reaction. We'll spend more time talking about this if you take a biochemistry course, but it's a condensation. It's technically a phosphodiester condensation reaction here. So we're forming this phospho, because it's phosphate, diester, because we're having an ester formation. And so it's a condensation reaction, so we're releasing water here. And here we've got our phosphate. So here, remember here, we're going to tack on a new nucleotide. So let's imagine we're that. So here's our original one. There's our base, there's our ribose, and there's our 5' phosphate. And we're going to bring in a new nucleotide. There's its 5' phosphate here. And what we're going to do is we're going to take this hydrogen here and this hydroxyl group here, and we're going to pull them off, make a water, and then form a new bond between the phosphate here and this 3'. So this oxygen binds to this phosphate. So this oxygen goes away and it gets replaced with this one. So we form a nice little bond there. Pretty great phosphodiester linkage, really, really important. It's pretty strong, which is nice as far as covalent bonds go. The hydrogen bonds actually between DNA is not as strong, which is going to come up later. So anyway, that's what I'm going to start referring to as our primary structure is just the order of nucleic acids in either DNA or RNA. Now, there is some nomenclature issues when we talk about primary, secondary, tertiary structures and quaternary structures with nucleic acids. So what you start to run into is there's some misnomers. With proteins, it's pretty straightforward. Primary is just the order of amino acids. Secondary is when you start getting some basic folds. Tertiary is when you start folding the folds. And then quaternary is when you start folding proteins together. Now, there are some subcategories I skipped over too. When you take a biochemistry course, that's what we'll talk about. Now, DNA, nucleic acids in general, also have a nomenclature that we sort of use. Like primary is the order of nucleic acids, just like for proteins, the order of amino acids is its primary structure. But once we get from primary to secondary, that's fine for DNA and RNA, but then it gets a little wonky when we start getting larger chunks, and especially with RNA. When we start combining RNA with other things, it gets confusing. But I wanted to just say that out loud because I'm going to start saying secondary and tertiary, and I might skip over some things, and it might be like, why did he skip that? Well, that's why, because it gets a little wonky, but whatever. So let's talk about DNA secondary structure. So generally, we consider secondary structure is when we start actually forming what will become our double helix. Okay, and that's where we start getting our hydrogen bonding between our guanines and our cytosines. We get 3 of those. And between our thymines and our adenines, that's where we have 2 hydrogen bonds. And then we've got our nice 5' to 3'. Notice the directionality here. 5' to 3' going in opposite directions. But it's still a nice sugar phosphate backbone. Now, something I wanted to hit, there's 2 big things here. Notice the structural differences. So we're matching up a purine, which is a guanine. Guanine is a purine, that's a type of nucleotide. So guanines, see they've got 2 rings here. And adenine, also 2 rings, so those are purines. And then we've got cytosine and thymine, and then uracil isn't here, but you could just block off this little methyl group, and that'd be a uracil. Those are our pyrimidines, and it's just one single ring structure here. Okay, so 2 rings is a purine, 1 ring is a pyrimidine. And we're going to match them up. So we combine a purine guanine with a pyrimidine cytosine, and that forms 3 hydrogen bonds. Perfect. And then we take to be consistent, we take our thymine, which is a pyrimidine, match it up to our adenine, which is a purine. Whoop-de-doo, but we only have 2 hydrogen bonds. It's fine. That's what's going to hold these 2 strands together. Now, that's a ton of hydrogen bonds. Just think about it. These are huge. DNA molecules are so long. So you get tons of these hydrogen bonds. Now, as we know, hydrogen bonds are way easier to break than a covalent bond, but they still have a pretty decent strength to them. So it takes some effort to pull them apart, which is really good. You don't want DNA just randomly falling apart unless you heat it up enough to do it, like if you were doing PCR, or you can do it mechanically by using a polymerase. And polymerazers have to open up that strand. We'll talk about that. It's more of a polymerase complex, but we'll get to that when we actually talk about replication and transcription. So those are different lectures. Now, the other thing I wanted to mention is sometimes you'll see in certain organisms, so with bacteria, sometimes we try and categorize things, right? That's what humans do, we categorize stuff. And one of the categories that we have for microorganisms are high GC content bacteria. There's entire subgroups of bacteria that we can define as saying high GC content. Well, what does that mean? Well, now we know. Guanine and cytosine, so they have high GC content. So if you have a high GC content bacteria, that just means that its DNA strands tend to have a lot of G's and C's. Well, what does that mean? Well, practically that means they have an extra hydrogen bond for a bunch of these nucleotides compared to maybe a bacteria that has, maybe let's say we have one bacteria that has 50% T's and A's and 50% G's and C's. All right, well, that's fine. There's some specific number of hydrogen bonds between those. Well, let's say we up our GCs to 75% and our T's and A's drop down to 25%. Well, there's going to be a lot tighter, tighter isn't the right word, there's going to be a lot more strength holding those two DNA strands together. And that's just classes of bacteria are categorized that way, which is just really, really interesting. So they're harder to pull apart. That also comes up with, if you're trying to do polymerase chain reaction, which we'll talk about at a later date, but that's just amplifying DNA, CSI kind of stuff, or if you need to do a quick test. So the coronavirus is happening right now. Some of the tests are PCR based, and that means you need to amplify DNA. Well, in order to do that, you need to understand the GC content of the region you're trying to amplify. If it's really, really high, that means that you need to heat up that DNA to actually separate it. You have to break those hydrogen bonds with extra heat. If you have low GC content, well, then you don't have to use as much heat. So just something to think about. As we move on to our tertiary structures, so we had primary is just the order of our nucleic acids. Secondary is when we combine these two strands together, right? 5' to 3', 5' to 3'. Tertiary structure is when we actually start getting some folding, and that's where it gets a little wonky, where tertiary structure, is it tertiary? Is it still sort of secondary? I don't know. I just threw it on here because that's what I kind of heard it listed as, but how we define that doesn't really matter. It's more important that you understand what's going on. So when we have these two strands going past each other in these two directions, we actually end up, it's a double helix. A lot of drawings of DNA are incorrect. They will show a single helix wrapped around itself. DNA is a double helix, so you should have two grooves, not one groove. So if it all looks like one exact same length between all of these twisties, then that is then that's not correct. There needs to be a small groove, a minor groove, and a major groove. Actual lengths of these isn't necessarily important for this point in this class. If you dig into more biochemistry and some of the details behind the actual structure of molecules, sure, you can get into that kind of stuff. But what's more important to me is understanding the purpose of having two grooves, major and minor. Generally, the major groove is where you're going to get DNA binding proteins. That's how they're going to access the nucleotides, because they need to be able to access the nucleotides. A lot of DNA binding proteins need access to the nucleotides so that they can do specific cutting or specific splicing. Some DNA binding proteins don't need that kind of issue, but they just need like a way to get access to the nucleotides. And so that's why you have two groups, major and minor. Now, we're going to keep on going a little bit deeper into DNA, and we're going to get to RNA in a second. So DNA can have further structures. Now, the B form, I've got three forms here. There's a couple other forms, but honestly, A, B, and Z are the three biggies. Really, B and A are the two bigger ones. But B form is the form that we generally think about. So it's the most common. It is right-handed. So if you can imagine, if you kind of look at it straight on, as we are, so I'm looking at it straight on, and you look at it and you're like, okay, the way I look at it is, if it's a spiral staircase, which way is it going? Well, it's kind of going up to the right, right? So you would be going counterclockwise, I guess, if you looked straight down. So that means it's right-handed because it's kind of going up to the right. Compare that to the Z form, and you can see it's actually going up to the left, right? And so that means it's left-handed. But anyway, let's talk a little bit about this. So the B form is the most common form. It's right-handed. You have a major groove and a minor groove, right? So you can see you have much more access to these gray nucleotides through the major groove than the minor groove. Now the A form is basically a dehydrated condition for DNA, still DNA. You just can squeeze more nucleotides in there. You can generate it under dehydration conditions, but you also see it in some of the weird hybrids that we see. So like RNA-RNA hybrids or DNA-RNA hybrids, which tend to more show up like in viruses as opposed to humans. But it's tighter. You can fit more nucleotides per turn of the DNA, which, I don't know, it's a thing. It obviously happens. These are the more naturally occurring ones, though B form is obviously the vast majority. Z form is kind of weird. It's poorly understood, but it's generally associated with a few diseases. You can find Z form more often. So it's almost like the entire molecule kind of got like shifted. If you grab the left side and the right side of the molecule and just kind of shifted them, and then it bends differently, and now it twists differently. It is associated with the rate of transcription in some diseases. But for the most part, honestly, the B form is the most important one. Unless you're really looking for the Z or A form, then maybe you'll find them. There are a couple other random ones, but they're relatively uncommon, especially in natural systems. You can get them to form, but yeah, they're pretty uncommon. Now, let's talk about RNA a little bit. So here's our DNA. It's just a different picture of our typical DNA double helix. Now, RNA, there's a bunch of varieties of RNA, but generally when people say RNA, they're usually talking about messenger RNA. Now, messenger RNA is what's going to deliver the sequence of nucleotides from DNA. We're going to sort of copy it into our single-stranded piece of RNA, and then we're going to send that to a ribosome to get translated. Now, it's usually single-stranded, but the trick with messenger RNA, or any of the RNAs actually, is that they can fold over on themselves. So they have these really cool folds that start to happen. It's kind of crazy. I have some pictures here. So you can see here's some folding. So you do get, we still have traditional bonding patterns. So you still have that triple hydrogen bond between, that's probably not a good way to say that. There's 3 hydrogen bonds, not a triple bond. There's 3 hydrogen bonds between guanine and cytosine still. There's 2 between adenine and uracil, right? Because it used to be thymine, but we transcribed it, and so now it's uracil. Because we need to be able to tell the difference between RNA and DNA. But as we go over here, so we still have that classic bonding, but we can get these folded over. So it starts looking weird. There's not 2 strands. It's one continuous strand. It looks like one of those weird ribbons, those infinity ribbon looking things. There's lots of different ways to study this. Oh, I don't have the reference here. I'll put that somewhere. Hang on, it's from Matthew's lab. They do a bunch of RNA work. So you can get all kinds of crazy folding. And when we dig a little bit deeper into RNA and biochemistry with nucleic acids, we'll dig deeper. If you take biochemistry, I will have lectures for that. If you don't take biochemistry with me, that's fine. Hopefully you'll talk about RNA at some point, or you can just Google it, because that's what the internet is for. You can get these interesting folds. And that's how we get deeper structure into our RNA. Now, is this tertiary or not? That's a good question. That's where the secondary versus tertiary versus quaternary, that gets a little wonky with RNA. But for the most part, just who cares? There can be folding of RNA, and that provides a lot of structure, and that can do all kinds of cool stuff. I was doing some work where we were using RNA folded over, and then if you fold RNA properly, which you kind of do it randomly, you'd take a chunk of RNA and you would fold it as many ways as you could. You'd have a bunch of those, and then you'd see which ones would bind to your protein. And so you would throw a bunch of proteins in there, and then you would do a pull down assay and see if any were bound. And then that becomes a delivery system. So like we're talking aptamers here. We were doing an aptamer delivery study of a gene therapy, which was pretty cool. So we were trying to treat HIV at the time. And so you can use these secondary structures to give you binding sites. So it's not just about the order of nucleic acids. That's really important. Okay? Sometimes that's all it's really about. But that secondary and tertiary structure of RNA is arguably just as important as the order of the nucleic acids. Okay? If the order is number 1, then number 2 is how it's folded. Because if you start misfolding things, you can really mess stuff up. So we'll talk a little bit more about that later too. Now, I mentioned here that RNA comes in many varieties. Now, messenger RNA is the common one. That's the one we talk about a lot. So I borrowed this from somebody. They put a bunch of stuff on here on the length. So length in nucleotides. So your messenger RNAs can be super long. they can be kind of short, but for the most part, they're going to be pretty long. Same with the ribosomal RNAs, which I'll talk about in a second. But you can have a bunch of these I'd never even heard of before. So transfer RNA is pretty small. That's the more common one on here. MicroRNAs and siRNAs. I think it's like small nuclear something RNAs. I don't know what piRNA is. There's so many, but you can see we usually just think about this one right here, or even these guys, and they're all really long. That's cool. But you can have all these non-coding RNAs and just crazy stuff, so like small non-coding RNA with different lengths, really cool. And that's where you start getting into some of the cool things that RNA can do. Like we'll use RNA as to guide your RISC complex to actually do splicing. RNA is a huge component of like ribosomes. So we'll talk about that in a second. Let's jump back to DNA really fast. So instead of saying tertiary structure for DNA, I just said higher order structures because we need to kind of talk about how DNA is organized, and there's lots of levels of organization. It needs to be very carefully organized, regardless of the organism that you are. If you're a higher order organism like humans, other eukaryotes, then you're going to have really big, high-level structures. You have a ton of stuff that you need to have. What do humans have? Like 20,000 genes or something like that? So you need to be nice and organized. And so that's where we get all our chromosomes. And so we take our DNA strand, our double helix, and we wrap it around these proteins called histones. There's a few different varieties of histones. So you can see in this one we have there's like the circle kind, the spheres, and attached to an H1. And then those get wrapped around each other. So you have these histone units, or the nucleosome subcomponents, and then the histones get wrapped around each other, and then you wrap the wraps around each other, and then you wrap those things, you start getting these really big, you get these long fibers of histones in DNA, and then those get looped around each other, and then eventually that's, if you condense it down during metaphase, because usually these are just kind of like this, they're just floating around in the nucleus, just everywhere. Polymerases are binding and unbinding, but when you go through mitosis, which will be a later lecture, then you actually get condensation. That's why I like this picture a lot, because it says metaphase chromosome. And so the metaphase chromosome is actually when you get this condensation. And there's a sister chromatid. You wouldn't normally see it condensed like this, and you wouldn't normally see two of these arms unless you're undergoing mitosis. So lots of higher order structure. We'll talk more at a later lecture about actual chromosomes and some of the chromosomal abnormalities that can happen. So stay tuned for that. RNA structure. So I mentioned ribosomes. There's so many of these. I had to just pick, I had all these other pictures on here, and I was like, oh man, I just have to leave this behind. We're going to talk more about RNA later, but there's some really cool stuff. So RNA, we can use it as a base component. It's not just messenger RNA. I mean, that's so important because that's how we actually make proteins. But there's other types of RNA, hence all of these crazy things. Now, one of the cool things is, so transfer RNA is going to come up when we talk about translation. This is what bridges the gap between, so it says anticodon here, so that's going to bridge the gap between a messenger RNA. So you're going to have your messenger RNA going along here, and it binds to this transfer RNA, which on this end of the transfer RNA is an amino acid, which is, we're going to order a bunch of those together to make a protein. And that's where proteins come from. So cool. Now, so that's transfer RNA. That's just one type of RNA. There's also ribosomal RNA. And I found this cool picture from a journal article. And they talk about the different components. So here's yeast, and here's a bacteria. Is that an archaea? Anyway, it's small. And so they're trying to compare different ribosomes. And but what I was really caring about was the fact that all this orange stuff here is RNA. All this purple stuff and this red chunk is RNA. Over here, there's just a different creature's ribosome, and all that yellow stuff, all the orange stuff, and then there's also that red stuff here. That's all RNA. So a lot of times people will say, oh, proteins are the things that are the machines of the cell, that are enzymes and that do stuff. Well, ribosomes, like ribosomes, are arguably more RNA than they are protein, which is really, really cool. There's 2 subunits, just to get this in there. We're going to talk way more about ribosomes and the actual process of translation later, but just to kind of get it in your heads a little bit, I have a little animation going on here. So here's my messenger RNA. It's going to bind right here. It binds in between the large subunit and the small subunit. Something else too, just while it's on my brain, we have 2 ribosomes. You can see we share units. So the small subunit has these 2 pieces. These are just ribosome proteins. And then in this other organism, we also have a small subunit, also have a large subunit, and there's the exact same chunks. Okay? The sizes can change. So the 18S versus 16S, that's sedimentation rate. It's sort of an analog for mass, but it has to do with how it actually forms like layers. Not really important. You can just substitute it for mass for now. It's just the problem is you could be like, oh, how big is a ribosome? Well, it's 23 plus 16. So that's what, 39, but it's actually not that size. It's probably like 35 acids, which is just confusing. So it confuses people. Anyway, so you've got your large subunit, you've got your small subunit, and here's your messenger RNA. It sticks in right here. It came out of the nucleus. And then our transfer RNA is going to come over here. Binds here. There's my amino acid. Sorry if it's hard to see right there. It attaches amino acid, and then the amino acid stays behind. The rest of the transfer RNA leaves. Ooh. And then my terrible animation. And then a new transfer RNA comes in, attaches a new amino acid, and then it eventually gets knocked off. And then we keep building these amino acids into a big long chain. And it pops out, and that's where we get proteins from. Very, very cool stuff. So let's just kind of end off here with how I view DNA and RNA and ribosomes and all that stuff. So, and want to talk about the central dogma, because that's very, very important. I'm probably going to talk about that in every single lecture for this course. So DNA, deoxyribonucleic acid, it stores nucleic acids. Okay, awesome. It's structurally stable. Awesome. We need that. You don't want your master blueprints of whatever you're trying to create or make to fall apart, right? So it needs to be stable, and it maintains the base order. So the actual order of G's and C's and T's and A's is so, so important. You start swapping those things, then down the line, your amino acid chain is going to be different. Okay, and we're going to talk more about that. I'll have a whole lecture on mutations and mutation repair. And that's what happens. You get a mutation, that means you're messing up that base order, and that's going to screw up everything down the line. So we don't want that to happen. So DNA is just a bunch of nucleic acids in a very specific order that's maintained and is stable. Now, when we actually perform transcription of that, that means I need to make a protein. And in order to make a protein, I need a photocopy of my master blueprint. So that's what messenger RNA is, or mRNA. It's still maintaining that order of bases. That's so important. So that's why I keep saying the order of the nucleic acid bases is really, really important. Now it's complementary, okay, which we'll get to later. So you can see if this strand on the left here is the strand that I actually want to make a protein from, then my RNA is going to be complementary to this other side of the DNA. We'll talk more about that later. It gets a little confusing. I get confused, so don't worry if it doesn't make sense. Anyway, so it's complementary. And then we send that RNA off to be translated. And so that's the process of transcription. So DNA going to RNA, and then, so that's transcription, and then going from RNA to a protein with a ribosome, that's called translation. So, and that's the central dogma of molecular biology, is going from DNA to RNA to protein. Now, there's other things in there. So there's a couple other RNAs in there. So we need transfer RNA to go from a messenger RNA to the actual amino acid chain. So that's what I talked about here. That's what this guy is. So I just wanted to mention that. It bridges that gap between messenger RNA and amino acids. I just wanted to mention, I just talked about it a bunch, but that there's ribosome RNA. So it's just a huge chunk of these ribosomes. But we're talking about RNA, so we need to mention that. If you're going to say the function of RNA is to act as a template for ribosome translation into a protein, that's an incorrect, it's not incorrect, It's an incomplete statement. You need to specify what type of RNA. Now, genes come up, and that's something people have heard of. Oh, I got good genes. I got them at Old Navy. No, I have genes. So what is a gene? Well, lots of people will say, oh, it's something that makes a protein. Okay, well, there's genes that don't do that. So, that's true in some cases, not always true in all cases. The best way that I think about it is it's the order of bases that can be transcribed. So it's a chunk of DNA that has a specific order of bases, a number of bases, that can be transcribed into an RNA and then translated into a set of amino acids that serves a function. Okay. Now, you could also argue that there are non-coding portions of DNA, and you could organize those into chunks, and you could argue that those are also genes. But it's basically like, if DNA is maintaining base order, well, if I start narrowing it down, like, oh, this chunk of 400 nucleic acids in this DNA forms this protein when I go via RNA. All right, fine, there's your gene. And that's how we start organizing them. It's more, we take a protein and kind of go backwards, and that's how we usually will categorize what a gene actually is. So, the central dogma, though, going from DNA to RNA to protein. I apologize if you hear that. A mail truck just drove by. Now, a couple things that I wanted to hit, and then we're going to be done. So DNA to RNA to protein, that's the central dogma. You can go backwards. That only really happens with retroviruses. So HIV being the most commonly understood one. There are other retroviruses, though. Baltimore class 6, I believe. And so we can transcribe and go from DNA to RNA. Awesome. Usually we go from RNA, well, messenger RNA, to a protein. That's translation. Now, sometimes, if you're a virus like HIV, you have an RNA genome. So instead of having DNA like we do, its genome is RNA, which is kind of weird, right? Well, it sends it backwards. So it will reverse transcribe or perform a reverse transcription using an enzyme called reverse transcriptase, ASE meaning an enzyme. And it turns its RNA into first an RNA-DNA hybrid and then a DNA-DNA. So a double-stranded DNA. And then that gets embedded in you, inside your cells, inside your genome, and then it's stuck there forever. Anyway, so that's transcription to translation, but you can also have reverse transcription. Very cool. Is there reverse translation? Not that I've seen. That's always up for debate. Could you go from a protein to an RNA? If you had the right enzyme, sure, but it's not something that really happens. The one other thing, too, I want to mention is replication. So when a cell needs to grow and divide and make new daughter cells, then it needs to double its DNA, because each of the new 2 daughter cells that are coming out of that first cell need to have 2 copies of the DNA, and it needs to be identical. So replication of that DNA is really, really important. And I have a little thing here I'm going to drag up here. These are the enzymes that perform that. So if I have my DNA strand and I need to make more DNA, I'm going to do replication using a DNA, right, because it's DNA, polymerase. I want to make more DNA. The polymerases refer to what they make. So if I have a DNA polymerase, it makes more DNA. If I have an RNA polymerase, it makes RNA. Reverse transcriptase is goofy. It takes RNA and turns it into DNA. So it's an RNA-dependent DNA polymerase, okay, is the more accurate term. Most RNA polymerases are DNA-dependent RNA polymerases, so it's kind of the opposite. DNA polymerases, there's a few different types. We'll talk about those later, but they're generally DNA-dependent DNA polymerases. Hopefully that kind of makes sense. We'll talk more about it later in a different lecture. And then you get your ribosome, which is an enzyme. Depends on how you define enzymes. It lowers the activation energy for actually performing this job, so I suppose. We generally call them ribosomes. And they're the ones who actually translate. So if I was going to ask you, I'd like to give questions. So great question to ask if you're taking this class with me or someone else. Kind of questions I ask, write out the central dogma in as much detail as you possibly can. I've had some really detailed drawings. So yeah, I'm at 40 minutes, so I'm going to stop here. Yeah, I'll talk to you later.